So this is going to be a plenary that's going to introduce you, and hopefully you can see everyone here. Oh, here I am on the screen too. Outstanding. So I'm super excited to introduce three wonderful people that I've had a pleasure to work with in the SMD, and we wanted to really introduce the ASGSR community to SMD and the different programs that are available within that, because I see a lot of potential for dovetailing a lot of what you guys do with the astrobiology, habitable worlds, planetary protection. I just see a lot of overlap. So when we were designing the meeting and designing the plenary, we wanted to really, now that we're in a new family, the SMD family, we wanted to really educate as many people as we can from the ASGSR community about what SMD really does. So that's kind of the point of the meeting, and I'm very pleased. I'm going to introduce all three of our panelists, and then they're going to trade off with a presentation to help kind of make you aware. And as always, if you have questions for any of the speakers, you can text me at 855-855-1413 for all you IT crowd fans out there. But so our first panelist I'll introduce is Dr. Michael New. He's the Deputy Associate Administrator for Research within the Science Mission Directorate at NASA. Of his many roles and responsibilities, he oversees the scientific competition process for research and flight awards in SMD. He mediates the relationship between the National Research Council and SMD, as well as coordinate several suborbital flight programs. He's a busy guy, and he manages all these and coordinates all these efforts. So prior to this role, he was the astrobiology discipline scientist and the lead scientist for the Discovery program in Planetary Science Division of SMD. And he got his PhD in chemical physics from Yale and is an Eagle Scout, which I know quite a few of you out there are Eagle Scouts. So I think that is a very cool and important thing. Dr. Lindsay Hayes is a program scientist. Maybe you can wave, Lindsay, so we know which one is. There you are, Lindsay. Great, you can hear me. So Dr. Lindsay Hayes is a program scientist in Planetary Science Division at NASA headquarters, and she's also the deputy program scientist for the Mars Sample Return Challenge, which is part of the 2020 Perseverance rover, and the deputy program scientist for the astrobiology program, where she leads, and she does it excellently, the exobiology research program and the finest graduate program. And she's also the NASA headquarters representative for the Prebiotic Chemistry and Early Earth Environments Research Coordination Network. And if you don't know what those are, feel free to reach out to me. Those are really important new opportunities for people to participate in different kinds of astrobiology research. So her scientific background is in biosignatures, and she really wrote one of the most fantastic review papers on biosignatures in astrobiology recently. And then there's Dr. Becky McCauley Ranch, who's also participating. She's a program scientist in the Planetary Science Division in SMD, and which is also at NASA headquarters. And she leads the Planetary Protection Research Program, which is really about helping reduce forward and reverse contamination of microbes. She also co-leads the Habitable Worlds program, which is helping to understand what is the criteria that makes a world habitable, and it's also part of the astrobiology program. And she's also a program scientist for Planetary Data System and is a NASA liaison for the MAPSIT program. And additionally, she's the deputy program scientist for the Curiosity Mars rover. So a lot of expertise from the SMD program is here in this panel. And so I'm going to turn it over to our panelists, and they'll talk a little bit more about the various different types of science in SMD. Who wants to, Lindsay, are you going first, right? Okay.
I think Michael's going first.
Oh, Michael's going to go first, sorry.
No worries, no worries. Hi, everybody, I'm Michael New. As Jamie said, I am the Deputy Associate Administrator for Research. Many moons ago, I was the astrobiology discipline scientist, so I ran the exobiology program, which Lindsay Hayes now runs quite admirably. And in fact, I funded Jamie at one point in time. So, old home week. So I'm here to talk to you a little bit about SMD kind of large scale. Lindsay, can you put up that slide you had with astrobiology and the divisions and everything? I think it's the last one in your package. Great. So this is kind of a family portrait of what the Science Mission Directorate looks like. We have 5 science divisions now: Astrophysics, Biological and Physical Sciences, Earth Science Division, Heliophysics, and Planetary Science Division. The Planetary Science Division is currently the home of the exobiology program, as well as the astrobiology program writ large. And Lindsay will tell you all about the parts and pieces of the astrobiology program in a minute or two. I'm very, very happy and excited to see its 5 divisions now, not 4, with the addition of biological and physical sciences. This is to a certain extent going forward to the past, since way back when we all used to be together in what was called the Office of Space Science and Applications, way before I ever got to NASA. You can take down the slide now. Thank you. So what's at the core of SMD? Core of SMD is transformative science. And we have 2 sort of pyramids, if you will, that we kind of build the base for our science on. The first is a diversity of career stages. So we have programs that go from graduate support, this is something called Finest, all the way up to our full-on grants programs. And Becky and Lindsay will be talking about those grants programs. We also have the NASA Postdoctoral Program Fellowship. This is a postdoctoral fellowship, and I know that the biological and physical sciences have had some, and they will continue to have more as time goes on. Right now, SMD sponsors about 125 of these. By sponsors, I mean funds, at 125. And there are maybe a half dozen that are currently done by the biological and physical sciences using funding from when they were part of HEO. Those slots will transfer over to SMD, and we are about to actually go through and figure out what the right number of slots is for each of the divisions. These postdoctoral fellows are free to you. They are mostly at NASA centers, however, although there are a few at non-NASA centers. But so building this pyramid, building this pipeline from early career researchers through all the way up to senior researchers is a big part of what SMD does, and it's a big part of how we think about our programs. I know that after 2005, when there were these tremendous cuts to the biological and physical research community, some of that pipeline, if you will, faltered a little bit. And so it's definitely one of our goals in SMD to build that back up again so that we have a healthy continuity of career stages. The second pyramid it's kind of based on is the idea that we build from laboratory, ground-based, or data analysis programs all the way up to missions, flight projects, with all kinds of stuff in between, technology development and flight demonstrations all the way. That's another pyramid that I think has also suffered a lot in biological and physical sciences since the 2005 cuts. And I think moving biological and physical sciences into SMD is going to help us reinvigorate that as well as the pipeline. Because again, this is a key way that SMD operates. We do stuff in the lab, we do stuff in the field, we do things on computers in preparation for or to make the most of our future missions. Now, of course, biological and physical sciences, until recently, doesn't really have an equivalent to a mission. So a NASA mission, an SMD mission is anything from a $30 million instrument that's sitting on the outside of the space station all the way to a $1 billion mission that's winging its way to Pluto. And until now, that hasn't been a big part of the biological and physical sciences community's methodology. You guys have two great platforms. You had the shuttle and the station. Now you have station that provides you with a whole bunch of facilities, and you have a process for making use of those facilities. And one of the things we're really hoping will come out of your newest decadal survey, which just kicked off, by the way, is a concept of campaigns. These are going to be extended in time, the equivalent of a spaceflight mission. And they may or may not, they may involve, equipment on station, they may involve facilities on Gateway, they may involve facilities on the surface of the moon or in orbit around the moon. But it can also take advantage, those campaigns can also take advantage of SMD's broader expertise in what you would call free flyers, what we call missions. So, your white papers, I think, are due for your decadal survey coming up. I think it's in December for the campaigns. And, personally, I'd like to see everybody put out some really wild ideas, submit ideas about how to do the science. And we'll figure out then how to find the resources to do that science. In other words, we'd really love to see creative ideas about, here's a science problem, here's how we're going to address it, and oh, by the way, we need a spacecraft that can be in orbit around Mars for 2 years to do it. Not, well, we know we're going to have this facility in this place, so here's some science we can do with it. So we do want you to think big, and that's the third part of the basis of SMD science, which is, thinking about what's transformative, what we need to do that transformative science, and then we go out and turn it into the resources we need to build those things. So I'm really excited again to have biological and physical sciences in SMD. I think we'll be able to do some great science and have some great collaborations as well. And I guess I'm willing to answer questions when we get to it. Thank you.
Thank you, Dr. New. And yes, just to reiterate the number, if you do have a question, if you could identify who you'd like to ask the question of, the phone number is 855-855-1413, and this is for the people streaming at home as well, watching the live stream. So I guess we'll go to Dr. Becky McCullough-Ranch, who will present her work, or her vision, or her component of the SMD project.
Yeah, I just wanted to check with Michael and make sure you didn't have a slide that you wanted to share at this point, right?
But we see it in presentation mode, so you might want to.
Let me switch the screens.
Perfect.
How is that? All right, great. All right, well, thank you so much for having us here today. It's really great. Sorry we're not there in person, but really just excited to be able to share the experience with everyone. So thank you so much for the intro earlier. While I wear many hats, I'm here today to talk about planetary protection and our research program. I actually do a lot of things with Lindsay as well in the astrobiology program, but I'm going to leave her to talk about all of those things. So first off, what I'd like to do is just give you a little bit of who's who in planetary protection at NASA headquarters. I especially think this comes up a lot because people are always so excited about planetary protection officers, which is not what my role is. So I put together this chart to kind of give folks a lay of the land. So what you can see here is that planetary protection is a cross-directorate activity, cross-office activity at NASA headquarters. Recently, in the past few years, we moved the Planetary Protection Officer from the Science Mission Directorate to the Office of Safety and Mission Assurance, which is actually an excellent location for that position. And that is currently filled by Nick Benardini, with Elaine Seasley as his deputy. And they are responsible for making sure that NASA and our missions are in compliance with all of those planetary protection requirements that we've agreed to through all of the various treaties and everything that we've put in place to make sure that we are protecting Earth, but also protecting all of the planets in the solar system. In the Science Mission Directorate, we still have several things related to planetary protection. It's a very important thing in what we do. And what I do is I manage our planetary protection research portfolio. So this is our opportunity in the Planetary Science Division for folks to submit proposals for research and technology development that will be mission enabling related to planetary protection, and making sure that we're continuing to push the way in which we meet those requirements, that we're doing so with the most up-to-date science, and that we're finding ways to make the process easier and easier for our mission teams to be able to meet those requirements. And so that's what that portfolio is, and I manage that. Ursula Rick is a policy analyst in the Science Mission Directorate, and she actually does a lot of things related to how we're working with other agencies and thinking about planetary protection, as well as a lot of connections that we have with the national academies, who has a committee on planetary protection that was set up in some recent years. And so she works a lot in that area. And then in our other mission directorates, what used to be HEOMD, but now is split into the Exploration Systems Development and Space Operations Mission Directorate, we have Julie Robinson and Betty Siegel, who are the Planetary Protection Integration Manager and Liaison, and I work with them both. We meet fairly regularly to talk about all of the things that are going on in our various mission directorates. And the same thing is true of Karen Knight over in the Space Technology Mission Directorate. So this is kind of the group that's really talking about planetary protection at NASA headquarters, and my role within that group. So with that, most people in this room, I think, probably already are familiar with planetary protection, but I'll just make sure we're all on the same playing field. So planetary protection is the practice of understanding and limiting terrestrial biological contamination, both on other solar system bodies, but also protecting Earth from possible life forms, especially as we think about sample return missions and things like that. And whenever we're doing that, we kind of break it into those two categories, right? So we think about forward contamination, which is us going out and exploring the solar system and what are we taking with us? How are we potentially contaminating things? And then the backward contamination, which is making sure that we don't bring anything back to Earth and introduce that to our ecosystem here. So the planetary protection research portfolio, which is what I manage, is that mission enabling, capability driven research that we really need to do in order to protect all of those solar system bodies. So we're focused on both forward and backward contamination, and we fund things that improve methods and technologies related to planetary protection, things that are going to detect contamination or remove contamination, or models that help us understand, what that's going to look like once something reaches its destination, or the risks that may be associated if something were to be off nominal. Our next solicitation is set to go out with Roses 22, so just stay tuned to Inspires, where we post all of our solicitations to learn more about that. Some recent activities that you all may be interested in. So as I mentioned earlier, the National Academies has a committee on planetary protection now, and there's actually been some recent reports out of that committee, specifically on lunar volatiles and bioburden requirements on Mars. So if those are topics that interest you, or you're just trying to understand what the current state of the field is in planetary protection, those are really great reports to go and look at. And stay tuned to that committee, because I'm sure that they'll be putting out more reports in the near future as well. This past month, we actually also selected 5 new awards out of our planetary protection research call. So those recently went out, and you can see the abstracts from those selections on Inspires if you want to get a sense of some of the awards that we're selecting. And as I mentioned earlier, Nick Benarodini is the planetary protection officer. He just moved into that role this past year. And I know that Lisa Pratt is actually, I think, giving one of your keynotes, I think, during your awards ceremony. And she was our planetary protection officer earlier this year. So I know there's a lot of text on this slide, but I thought, especially if I was going to talk about our planetary protection research portfolio, I wanted to give you all some sense of the types of projects that we're funding out of this. And so I listed the past 3 years where we've had selections. You can see the most recent ones, the titles there in Rose's 21. Some of the ones that I really want to draw your attention to, and I don't know, I'm guessing you can't see my cursor. I don't know.
Yes, we can.
Oh, you can. Okay, great. So we have, let's see. There is a lot on this slide. Yes, right here. Okay, so there's an EVA swab tool that we have been funding down at Johnson Space Center. And that's a really interesting work on figuring out ways to develop a tool to be able to take a sample with someone doing an EVA and kind of the difficulties associated with that and making sure that you can get a pristine sample up on the ISS. And so that's been really interesting work being done by Aaron Redberg at Johnson. And then I think the other one that I'll mention is right here, laser-based cleaning and in situ verification of bioloading. This is some work being done by Meg Abraham at Aerospace Corps. And it's really cool, and I think it's cool to your community as well because it's a lot about being able to detect microbes, very, very low levels, do some spectroscopy with them, and then be able to remove them. So just kind of thinking about that space environment and some of the things you might want to do. So I think that there's a lot of opportunity for overlap in terms of the expertise and the methodologies that are being applied across planetary protection and space biology. And so definitely some synergies there that folks could take some advantage of. And then last slide I have, I just wanted to, give a little bit of a plug for some of our planetary protection talks and posters that are going to be at this conference this week. And so just wanted to draw folks' attention to those. So Andy Spry is going to be talking tomorrow about microbiology and the deep space environment from the planetary protection perspective. And then Venkat is going to be giving a talk in, I think, the same session on characterizing spacesuit-associated microbial communities and their implications for future human missions. So that should be really interesting. And then, as I mentioned earlier, I believe Lisa Pratt, who is our former planetary protection officer, will be the speaker at your banquet and award ceremony, and we'll be talking about the 21st century space race and what that means for first contact. So those are some really great talks that those of you that may be interested in planetary protection might want to check out. And with that, I will pass it over to my other panelists.
Thank you, Becky. And again,
Am I sharing? Sorry, go ahead.
Sorry, just a plug. Again, if you have questions coming in, I don't have too many here, but 855-855-1413. Take it away, Lindsay.
Thank you so much, Jamie. I assume you guys can see the top slide. It says I'm screen sharing, but a nod. Fantastic. Thank you very much. Thank you guys for the introduction, Jamie and Michael and Becky, and setting us all up along this way. My name is Lindsay Hayes. As introduced, I'm the Deputy Program Scientist for the Astrobiology Program, and more broadly, just a Program Scientist in the Planetary Science Division. This is the slide that Michael also talked to and hinted that I would be talking to a bit as well. This is just to sort of give you a sense of the placement of the astrobiology program sort of within the Planetary Science Division, which is one of the 5 divisions that are part of the Science Mission Directorate. And as Becky was talking about, there are multiple directorates that have sort of related topics and research and such. So within the Science Mission Directorate is the Planetary Science Division. Within the Planetary Science Division is the primary home for the astrobiology program. Although, of course, it has ties to many of the other divisions, in fact, all of the other divisions, it has some ties to all of them. And then within the astrobiology program, there are a number of funding programs. Exobiology, Habitable Worlds, ICAR, P-STAR, Picasso, and Matisse are all different funding programs that we have to fund different aspects of astrobiology research. There are also research coordination networks, and these are, and as Jamie hinted at earlier, the PCE3 is just one of them. I'll talk a little bit more about a range of the research coordination networks that we have. And these are really more, those are sort of half within our sphere, half sort of community-based research networks for members of the community to come together. These are sort of the core members there are NASA-funded researchers, but they sort of bring together in specific topics a way to sort of coordinate different topical ideas, different folks doing research and sort of push the field forward in that way. And then there's a whole bunch of other things that are part of the astrobiology program that venture sort of even beyond that. There's astrobiology society activities we do. Michael mentioned there's NPPs, there's FINUS, which is our student program. There's just, there's a lot of different things that the astrobiology program does. And I'm just going to talk to you about a few of these as I go along. So broadly, of course, the field of astrobiology seeks to really address three main questions. How did we get here? Where are we going? And are we alone? And to answer these questions, astrobiology sort of at the topmost level looks to bring together knowledge of space environments and knowledge of Earth organisms to answer these questions. Now, at NASA, astrobiology has been part of NASA's science, part of our goals since 1963, which is very shortly after the agency was formed. So really, astrobiology has been a core part of NASA for quite a long time. And within NASA, the astrobiology science goals really focus on what are the characteristics of the solar system that lead to habitable environments, and how and where did or could life begin and evolve in the solar system or beyond. And so we lay these topics out in a very large document that we call the Astrobiology Science Strategy. This is sort of our roadmap, and it in fact has evolved from previous roadmaps that we had, previous astrobiology roadmaps. But this is really an expansion on that idea. This Astrobiology Science Strategy, which was released in fall 2015, is community-based and semi-decadal. Like I said, it was released in 2015. We're going to see as the planetary science decadal survey comes out soon, sort of where astrobiology is included in that, and use that as a sort of way to bring the astrobiology goals forward. But right now, this is our major driver, our major description of the field of astrobiology within the NASA context. One of the great things about the creation of the Astrobiology Science Strategy is that there was broad participation. Within the astrobiology program, Mary Wojtek, the senior scientist for astrobiology, and this was also, of course, when Michael New was the discipline scientist for astrobiology, and myself really worked to get broad participation across the science community in a wide range of fields related to astrobiology. And the goal was really to make as much of the science community that was part of astrobiology be able to see themselves somewhere in this document. The goal was to create an inspirational and aspirational document. One of the key features in each of the 6 major science chapters, which I'll talk a little bit about in the next slide, was where is this field going? Where do you want to see this field in 10 years? And the idea being, it's not just what people are doing now, but what are the questions that are driving us towards the next stage in research and in the field. I encourage you to take a look. There's a PDF version available on the astrobiology website, which is astrobiology.nasa.gov. As mentioned, the 6 major research areas covered in the strategy were identifying abiotic sources of organic compounds, really sort of where the organic compounds that sort of start to build to life, where they can be found, how they get there, that sort of thing. The synthesis and function of macromolecules, so going from sort of simple prebiotic compounds into more biologically driven macromolecules, the things that we think about as being necessary for sort of proto-life, early life. The next one is early life and increasing complexity, sort of what did the earliest forms of life look like? How did they start to become more complex? The coevolution of life and the physical environment. So as life began to really take a hold on this planet, how do we see evidence of life evolving to really have an effect on the physical environment, and how that environment had an effect on life, and how that has evolved through the 4.5 billion years of our planet's history? Identifying, exploring, and characterizing environments for habitability and biosignatures. And so that's starting to look more outwards, look more beyond our solar system and to different environments within our solar system, but really beyond the Earth. And then constructing habitable worlds. You know, if you want a planet that can support life, what does that look like? What do things need to go into it? So please note that the last two do have a lot of overlaps with the astrophysics directorate and a lot of the work that they have, or division, and a lot of the work that they do there as well. So I'm going to go a little bit into detail about each of these 6 major areas. And if you have questions, Jamie has given you the phone number multiple times, or we're going to have a question and answer section at the end. So if you ever want me to come back, please let me know. But to start with, abiotic sources of organic compounds. This really gets into where did the building blocks of life come from? How did these abiotic, these non-biological production of small molecules lead to sort of the production of large and more complex molecules? How does this lead to prebiotic chemistry and then the sort of origin of life on Earth? And this also encompasses the idea of did or could life or prebiotic chemistry evolve on differentiated altered icy worlds like Enceladus, Europa, Titan, etc. So this is just sort of a figure to bring up some of the text. Really, when we're thinking about this first topic, we're trying to get into questions of where did the ingredients of life originate? We think about sort of planetary system formation. Some material survived and was incorporated into small bodies, planetesimals, and then eventually delivered to planetary surfaces. So at the topmost level, a lot of those kinds of things we see and you can learn about from observations and simulations. The middle kind of stuff, understanding what's in or was in comets, interplanetary dust particles, asteroids, and planetesimals. We make those measurements with lab analyses and virtual samples or things that have meteorites, that sort of thing. And then the bottom, the sort of chemistry on the early Earth, a lot of that is also done through simulations. Moving into the second topic, the synthesis and function of macromolecules. This really gets to questions of what are the characteristics of the chemical processes that lead to the formation and persistence of the larger polymeric systems that are capable of the biologically important functions, the information, the sort of mechanisms of life. What are the interactions, intermediary structures and functions, energy sources, environmental factors, and how these contributed to the diversity, selection, and replication of these systems. So when we think about this, to get into less words, more pictures, thinking about it on a broader scale, we think about synthetic biology. And in the field of astrobiology, synthetic biology really focuses on building life from scratch, but not in the way of engineering organisms for a particular goal and applied function. We're not trying to find organisms that will do a particular thing. We're sort of doing a synthetic evolution kind of thing. This figures you see here is the evolution of oil droplets in a chemorobotic platform. There's a 3D printer which generates droplets and then sort of measures their fitness against a particularly defined fitness function, and then uses a genetic algorithm to generate a new population. And in that way, we can sort of see how this metabolism works and how this evolution works in a way that allows us to sort of understand different ways of synthetically biological evolution. The third topic is early life and increasing complexity. The questions here are things like, do overarching rules that govern evolutionary processes exist? And if they do, what are they? What are the processes that drive these big things? What are the attributes of life that are common to all origin scenarios or environments? And which are context dependent? Which are the things that we can expect to see on any planet that hosts life? And which are the things that are specific to our Earth? And how do we understand that? And this really also gets into this really important question of, is it a fruitful approach to try and define life? So the concept of universal biology, how to identify common features of life on Earth that can be extended beyond Earth. This is sort of a physicist view of biology that leads to an abstraction of function and features that can then be attributed to universal systems. So this is important for getting past this chicken or egg, which evolved first, metabolism or genetics, problem in biology, in order to apply biological concepts beyond the specific Earth example that we see. It's less about, was it genes or was it proteins, and more more, are information molecules and sort of process molecules both important for life? And, can they evolve separately? Do they always have to evolve together? And certainly, as we've seen by looking at the diversity of planets and systems within our solar system, every planet has its own story, its own history, and it's highly unlikely that the sort of specific details that we see on our planet would be replicated elsewhere. We need ways to understand life that can survive these sort of different types of environments that are more capital U universal. The fourth topic within our astrobiology science strategy is coevolution of life and the physical environment. And so these questions, as I was saying before, this really gets at what are the relationships between life and the physical environment in which it exists, right? The abundance of oxygen in our atmosphere today has certainly had an effect on weathering systems of the rocks and things like that. And those two things can provide a feedback. And how do those two things evolve together on not just a habitable world, but an inhabited world? How do the biological and abiogical parts of the world shape each other? And what evidence from Earth's coevolutionary past can inform our understanding of the habitability of Earth through time, which is a really important question because early Earth certainly looked like a different kind of habitable planet than our Earth does today. And also the habitability of environments in our solar system and beyond. This gets into questions related to paleogenetics and paleobiochemistry. The growth of bioinformatics databases has enabled detailed evidence-based predictions about the molecular biology of early life. And as really understanding of ancestral sequence reconstruction has allowed us to be a powerful tool for studying ancient cellular life. In some cases, we've been able to get ancestral metabolic pathways to be assembled from this data. And this has provided greater insight in metabolic capabilities of ancient organisms. So not just what life was, not just what those enzymes may have been like, but sort of exactly how different organisms, Luca or the last bacterial common ancestor, those sorts of things, how those organisms may have operated and how that may have turned into the phylogenetic trees that we see today. This also ties to questions about the earliest life on Earth, of course, and sort of understanding some of these. These are some of the oldest sedimentary rocks in the world, and that give us some of the earliest life some of the earliest evidence for life on Earth. And this ties to questions of, when did that oxygen rise that I was talking about, the oxygen that we see in abundance today, when did that start? How did that come about? What were some of the feedback loops that happened between an early low oxygen Earth and modern fully oxygenated surface? The fifth topic, to keep moving forwards inexorably, is environments for habitability and biosignatures. So these questions are really, how do we determine if a particular environment is currently habitable or was habitable in the past, is able to support the generation of life or was able to support that generation in the past? And how should we define habitability indicators like biosignatures and then interpret them in a context-sensitive manner? So this really gets to the broader questions, if this is what early Earth looked like, that certainly doesn't look like the habitable Earth that we're familiar with today, but how do we understand habitability on Earth and then search for similar conditions, but not necessarily the same conditions in environments and planets beyond Earth? And so the sixth and final topic that's part of this astrobiology science strategy is about making habitable worlds. So how has our limited experience with habitability, right? We have the one Earth, limited our basic understanding of the requirements for habitability, right? We know that things on this planet are required for habitability, but we don't necessarily know what the most simple set of those things are. And so how can we take our limited understanding of habitability and apply them to other environments within our solar system and beyond? And this gets it, as I mentioned, this has to do with some astrophysics related things. This is from a paper by Meadows and Barnes in 2018. And ultimately the sort of background to this is planets are hard. There's a lot of things that go into making a planet, just a planet. And then when you layer on top of that the things that are required for making a planet habitable, it's hard to create all of those things together. And how do we understand all of those things? It is hard to make a planet, it is hard to make a habitable planet, but things like Kepler have shown us that there are abundance of planets, beyond our solar system. And so how do we understand what is needed to create a habitable planet so we can start thinking about looking for habitability indicators and even in the future biosignatures on those planets? So I'm going to make a quick shift from sort of what is astrobiology science to what is funded in astrobiology science, especially at NASA. So I'm going to talk about 3 programs and then also the research coordination networks. I'll start with Habitable Worlds. Becky is actually the lead for the Habitable Worlds program. I'm her deputy for that. So the Habitable Worlds program really is focusing on research that uses the knowledge of the history of the Earth and the life on it to help determine processes, conditions that create and maintain habitable environments, and to search for ancient and contemporary environments and explore the possibility of extant life beyond the Earth, as well as the built environment. So this is one of the major changes we made recently in Habitable Worlds. This is also interested in looking at built environments on the space station or other places and understanding the habitability of those environments. So the research areas include, but are not limited to, the presence of water, exotic solvents, sources of energy for life, the presence of organics and their reactivity, water body physics, chemistry. You can read a lot of this. I encourage you, if this is something that you're interested in, to go look at the call. This particular program is solicited annually, although it is not being solicited in the Roses 21 call, so it won't be solicited until the Roses 22 call, which comes out in sort of mid-February. But this program typically selects about 5 to 10 proposals per year, depending on funds available and research projects and such. One of the other programs that NASA Astrobiology funds is the Exobiology program. As Michael mentioned, he's the former lead for the Exobiology program. I'm the current lead for the Exobiology program. This program, the aim is to understand the origin, evolution, distribution, and future of life in the universe. The research here is sort of centered, of course, on the origin and evolution of life, the potential of life to adapt to different environments, and the implications for life elsewhere. In some ways, you can sort of think of habitable worlds and exobiology as 2 sides of the coin. Really, habitable worlds is trying to understand the habitability of environments, and exobiology is really trying to understand how life lives in different environments. So those are the sort of balancing sides of that. The areas of research emphasis for exobiology include prebiotic evolution, early evolution of life in the biosphere, evolution of advanced life, large-scale environmental change and macroevolution, and biosignatures and life elsewhere. You can see a lot of the same themes that we were talking about in the science strategy that are reflected here. This program is solicited annually, so it's you can cut it every year, although we have shifted this program to what's known as the no due dates model. So if you've got a really great topic, you're excited about it, you can submit your proposal whenever it's ready to be, whenever it's completed, whenever it's ready to be solicited or submitted. And typically we select somewhere in the 20 to 25 proposals per year. Again, depending on that year's budget, what we currently are carrying in the program, other programmatic factors, that sort of thing. I'm going to step away from the funding for a moment to talk about the research coordination networks, and then I'm going to bring back the funding because they are related to that. So we have 5 astrobiology research coordination networks. The first of these was about exoplanet system science, which is the Nexus for exoplanet system science. The second one created was NFOLD, the Network for Life Detection. The third is really looking at prebiotic chemistry in the early Earth, which is PCE3, prebiotic chemistry in early Earth environments. Ocean Worlds is the topic of the fourth, which is the Network for Ocean Worlds. And the fifth and final one, earliest cells to multicellularity, it is in the process of being spun up at this moment, and Becky is actually the headquarters representative for that. I'm the headquarters representative for PCE3, and Mary Wojtek, the senior scientist for astrobiology, is the headquarters representative for the other 3. As I was saying earlier, the astrobiology research coordination networks, they are not funding mechanisms. The core teams in each of these RCNs are NASA-funded scientists who have applied to one of our other programs, either in PSD, some of them for example, some of those are funded out of the Earth Science Division. Some of the Nexus core teams are actually funded out of the Astrophysics Division. But these are NASA-funded scientists that make up the core of the team, but they're really create a broader community around the topics that you can see here and bring together scientists working on these topics to sort of sometimes topics that have generally been in sort of disparate fields, or sometimes topics where the communities don't necessarily talk. These are areas that within astrobiology we recognize that creating a structure around which groups can come together, different research groups can communicate their research and coordinate to new and interesting discoveries in fields of research, could really use that structure. So that's what we've built here. As I said, you know, we have 5 total. The 5th one is getting spun up. But one of the ways to sort of be thinking about these is that the ones that are going from the sort of northwest to the southeast are related to missions, right? The Network for Ocean Worlds is really thinking about what we know about our planets and the ocean worlds on it as a way to think about exploring ocean worlds outside the solar system. The Nexus is really thinking about outside of the solar system, exoplanets, and Enfold is thinking about life detection sort of wherever we may find it. Whereas across the other axis, PCE3 and early cells to multicellularity and some aspects of Enfold are really more about fundamental research, right? Understanding what happened on our planet as ways to understand what may be happening elsewhere. So the reason these RCNs are in the funding part of the presentation, even though they are not funding networks themselves, is that they feed into what we call the Interdisciplinary Consortium for Astrobiology Research. Now, this is for big proposals, right? These are proposals that describe a multi-million-dollar 5-year project with an interdisciplinary approach to a single compelling question. These are sort of larger than the scope of the individual research programs like exobiology and habitable worlds, but they do have to be related to one of those research coordination networks. And any teams that get selected out of this become a member of those. The calls occur on the order every 2 to 3 years. The last one was solicited in Rose's 19 and was linked to either Nexus, exoplanets, PCE3, sort of the prebiotic chemistry to the early Earth, or early cells of multicellularity. And the next call for this will be in Rose's 22. Now, I talked about 3 different research funding. I don't have a slick presentation or a slide like Becky had with the specific names of every single thing that's been selected. But if you're interested in seeing what types of research are there, this is the NSPIRES page. It can be found the same way that Becky referenced nspires.nasaprs.com. You can find it there. But anyways, on each of these for the past solicitations, you can go onto the page, you can see the actual call itself, but you can also see the selections, and that gives you a sense of the work that's been selected in the past. There's abstracts there, titles of the projects, those sorts of things, as a way to understand, you know, what research is being done, is being selected, is being funded. My final slide, I'm actually going to put up here, and I'm going to leave it up if that's okay. This is the slide that sort of shows, it sort of goes over the 3 different topics that we had with sort of space biology, you know, broader in SMD, with planetary protection, and astrobiology as a way to sort of look at the different types of research that are done, the similarities between the 3 and the overlaps between the 3, and the different things that sort of isolate them, I don't want to say from one another, but sort of define the different distinctions. And I think with that, I'm going to wrap up my portion. And Jamie, if this is a good time, I can leave this up or I can take this down, and we can get into some question and answer at your preference.
Can the audience read that okay? Is it a little small? I'm getting thumbs up. So, okay, we're going to leave it up there if that's okay. And we have a lot of questions that might dovetail into this last slide, actually. So thank you all. Can we give a round of applause for our plenary speakers? And again, the phone number for questions is 855-855-1413. So first of all, Lindsay, can you give an example of a selected proposal in the biosignatures area? Can you, I guess the audience is maybe a little unsure of what you might fund in that particular category.
You know, the first thing that comes to mind is actually a technosignatures proposal, although perhaps that's not a great example. Some of the work that we fund in the biosignatures field includes things like trying to understand biosignatures for particular types of metabolisms, or more broadly, biosignatures that get preserved. in deep time. So how may we understand the preservation of particular types of lipid biosignatures in different environments as a way to understand whether or not we could look for, this particular type of lipid on Mars. And so that's one of the things that we look for. I know that the term biosignatures has a very different definition in the medical field. And so really what we're looking for here is not signs of particular types of, we're not specific, we're not saying, is this metabolism necessarily present, but actually more broadly, is this a sign that life was here doing something at a particular time?
Great, thank you very much. This question is for Becky. Does planetary protection address the microbiome of the built environment or on the ISS, or what potential new contamination might be happening on Gateway or Orion as it's moving back and forth, say, from the moon or on station, or the Gateway station, especially if Gateway has long periods of uncrewed operation. So how does planetary protection fold into that?
Yeah, I mean, so what we're really interested in are going to be the protection of those planetary bodies, right? So if perhaps something was being collected on the Gateway as a break the chain sort of process, then we might be interested in kind of what's going on there. We're not that concerned with us taking the microbiome to the ISS, thinking about maybe what's happening, some of the changes that occur there, and then coming back to Earth. I mean, obviously it's a radiation environment, it's a little different, but it's still, the source is still the same, right? It's still that terrestrial microbe that is going back to a terrestrial environment. So we're not really that concerned about that. But what we are really interested in are potential planetary protection research and technology development that are things that can kind of happen with the ISS or Gateway. So for example, we're really interested to know how the space environment perhaps provides some sterilization to a spacecraft or to a mission along the way, right? And so having something out there that's unmanned for a certain period of time, that maybe there's some sort of microbes that have been left there, and being able to study how the radiation environment or just the space environment in general is decreasing that biological contamination. That's definitely some research we are interested in, and we've actually had a couple conversations about potential opportunistic research that might be able to be enabled by the Gateway or space stations.
Great. And this is, this has been touched on a little bit throughout the day, but maybe, Michael, you might touch upon this a little bit. The question was, what's the rationale for moving BPS into SMD? And Jim Green talked about it a little bit this morning. Craig kind of touched upon it. But I would love to hear, from the planetary side, about the move and maybe why that move happened, because I think a lot of people are excited about it, but maybe don't know how that came to be.
So I wasn't in any of the rooms where this was discussed, so I can't give you any, fly on the wall kind of secrets. What I can say is that SMD is the place where, until BPS came in, most of the basic research in NASA was being done. And with BPS coming into SMD, we could now say that almost all of the basic research being done at NASA is in one place. That allows us to learn from each other, to take the best practices of one group and apply them to another group. It allows us to cross-fertilize each other in terms of ideas, such as the question that they just asked Becky about the microbiology of the built environment. And it allows us to start thinking about breaking down some of these barriers to do interdivisional research. So work that maybe is relevant to both exoplanets and heliophysics, for example. So the work of looking at the effects of stars on the habitability of their planets and so on. So there's a lot of natural synergies that come about by having most of your science or almost all of your science in one place. Now, the science that's not in one place, of course, is the human research program, which I think over the last decade or so has taken on a little more of a applied role, figuring out what are the threats to humans in space and trying to develop countermeasures and protections for them. But most of the science is now in one place, and it gives us, like I said, all these positive ideas.
Great. Thank you very much. This goes back to Becky, kind of building on what you talked about before, about also involving the commercial landers, the CLIPS programs and research hardware. Are they complying with the NASA's planetary protection requirements, or how is that consultation done?
Yeah, so I'm not directly involved in those conversations. That would be the Planetary Protection Office. But from my interactions, I can tell you, those lines are open, those communication lines are open, and especially those payloads where you have NASA-funded payloads that are going to the moon. Planetary protection is, taking a front seat, and it's, getting approvals on their planetary protection plans from the Planetary Protection Officer, all in line with the way that we look at the moon and its categorization based off of what we understand about the habitability of the lunar environment. So I think that answers the question in general. Obviously, I think I'll just leave it there. I think that answers the question.
Yeah, thank you. The next question is for actually any of you, because you're all involved in this, but maybe it's directed mainly at Michael, is what are the opportunities for collaborative grants, like more cross-disciplinary, so that maybe part of it is habitable worlds and part of it is space biology or something like that. So do you see any kind of, do you expect unique funding lines, or do you expect any kind of cross-disciplinary opportunities for new funding for projects?
Yeah, that you've hit on a real interesting aspect. I think, Jamie, your mic is open because I can hear my feedback in your room.
Oh, really? Because
Yeah.
I'm asking the questions, so that's why I'm...
Right. When I'm talking, I hear the echo back. It doesn't matter.
I don't have the ability to mute myself, I'm sorry.
All right, well, we'll just have to deal. We will soldier on. So, developing ways to solicit and support interdivisional research is high on SMD's list of things we want to see. We have a couple of examples of programs that work that way. Probably the most integrated is something called Exoplanet Research, the Exoplanet Research Program, XRP. Their proposals are submitted to that program, and they are grouped into panels based purely on the science, and then they are evaluated based on their science. And it's only after the evaluations have been finished do we then decide which of the 4 divisions that are participating are going to fund which of the selected proposals. So that's sort of a hallmark benchmark program. We don't have one that is currently set up to be explicitly about biological and physical sciences, although that is clearly something we're going to be thinking about and talking about, not just with the 2 other biological programs you see on the slide, but there's also probably places where we could do some interdivisional research between the physical sciences side of BPS and either the astrophysics division or the heliophysics division, depending on what you're actually working on. To catalyze this, starting this coming year, we've created something called the Research Catalyst Fund. This is a very small pot of money. It's only $3 to $5 million, and it's administered in my office. And 3 to 4 times a year, we're going to ask all of the divisions to send us proposals that would otherwise not be funded because of funding issues. And in particular, these proposals would be in one of 3 types. Either they are proposals from minority serving or primarily undergraduate serving institutions. They are interdivisional research, meaning that they address the objectives of more than one of our science divisions, or they are what we consider to be high intellectual risk, high impact proposals that, and so the idea is to provide some co-funding to get them get these types of proposals into the funding range, get them started, and then move forward. That money is not coming out of anybody else's pocket. It is new money, but it is, as I said, a very small pot. It's only $3 to $5 million. So yeah, I think that was kind of a long-winded answer, I think, but.
No, that's great. And if I might add too, is with the graduate program Finest, is that also on the table too, that you guys are going to be expanding the graduate opportunities for space biologists and physical sciences?
So the way Finest works right now is each division chooses how many Finest awards it's going to make. It's not funded out of a central pot. So as budgets grow and contract over the years, the number of Finest awards grows and contracts over the years. But it is a centralized, it's not, the solicitation and evaluation is more or less centralized, but the individual funding is not. So I don't control that spigot the same way that I control the NPP program, the postdoctoral program.
Gotcha.
But yes, yeah.
Now, Lindsay, I did not write this question. You might, once you hear the question, you might think I wrote it, but this is either for Becky or Lindsay. It seems like detecting unknown or microbial dark matter would be important for planetary protection and space biology, as well as astrobiology. They wanted your thoughts and comments about efforts to look for microbial dark matter.
Right, I'll answer first, Becky, and then if you want to talk about planetary protection. The reason, of course, that Jamie thinks that I'm assuming that she wrote this question is that Jamie is actually funded by the exobiology program to study microbial dark matter. And so, yeah, it's definitely something, depending on the sort of purpose of your research and really trying to understand, you know, sort of the underlying science question of the particular research that you're trying to do. Understanding microbial dark matter, it can definitely be something that we consider relevant in the exobiology program. And you know, it is an important thing to understand, and it helps us understand more about the sort of universal features of life and how we detect life that may look different than what we're used to.
Yeah, and I guess what I'll add to that is, so in planetary protection, especially, well, okay, so with forward contamination, it's really easy, right? We have a pretty good understanding of the types of life forms we find on Earth and the ways that we can kind of mitigate those and find them. That's easy. Backward contamination has got a different issue, right? Which is, you know, we don't necessarily know maybe what we're looking for. And that is something that we're interested in the planetary protection program. And it's part of the reason we actually have one project looking at prions and how they may be degraded or deactivated by the space environment. And the reason we're interested in them in particular is that they're very hardy types of molecules. And so, you know, we're hoping that, you know, if we can feel good about that sort of decontamination, that we would feel good about things in that area. So anything in that kind of realm where we're thinking about like, okay, how would we you know, make sure that we're not bringing back something that we don't really totally understand as well as we understand Earth life. Those are things that we're interested in planetary protection, and not only from that kind of you know, prion perspective necessarily, or dark matter, but also in a process, right? You know, in just thinking about like, okay, well, what maybe are the ways that you mitigate that risk, and you know, can you do that through break the chain or some sort of more universal sterilization techniques that at least for all the life forms, as we know, and all the biologically active molecules that we can begin to think about, that we can feel very confident that they would be deactivated using those chemicals or those types of energy sources or whatever. So
So I have one more question. So if there's other questions that people have, please text to 855-855-1413. So the last question I have is kind of about data sharing and maybe a collaboration in sharing information, the long history that space biology's had and astrobiology's had. Is there any plans to share information, share data, I guess, for example, new microbiomes that are discovered in new environments or habitable worlds? Are there any efforts to have workshops or data sharing efforts, like beyond just depositing something in GeneLab or something like that? So that was a question from the audience about data sharing. And that can be to anyone.
I guess this is Michael. I guess I can address this. So as far as workshops go, we absolutely would have workshops. We have actually a proposal call called TWISC, Topical Workshop Symposia and Conferences. And if you want some support for putting together a workshop on something, you can propose to it. Not a dirty secret that most of the proposals that meet the requirements of that program do get funded. So we do have money available for that. Additionally, we have some great program officers. There are two of them on the panel right now, and they can also, you can talk to them about it and they can put together something. With regard to data sharing, you guys are really plugged into the trends within SMD, it seems, because one of the big things we are beginning to move into is open science, which includes not just sharing data, but also sharing metadata, data about data, and sharing our software. And so over the next coming years, we're going to be pushing that way in a big way. The first probably product from all of that you'll see is a SMD-wide data catalog that is being developed right now. And of course, since biological and physical sciences are now part of SMD, that catalog will include the GeneLab database as well as the physical informatics database. So yeah, and if you have ideas, bring them on down. We're happy to do it.
Well, thank you. Any last call for questions from the audience? Well, thank you all. Thank you, Michael, Lindsay, Becky, for your time today, and we really appreciate this warm welcome into the SMD family. So thank you very much.
Thank you for having us.
Thank you so much.
Well, we hope all of you will stick around and come to our reception. Each of you, I'll be emailing you a glass of wine. It will be virtual. So we appreciate you participating, so thank you. And I hope you all will come to the reception in about 20 minutes. Thank you.